and get started. So it is my pleasure today to introduce Dr. Paul Yager. Dr. Yager received his undergraduate degree in biochemistry from Princeton University and then went on to get his PhD in chemistry from the University of Oregon. After graduating, he worked at the Naval Research Laboratory in Washington, D.C. before moving out to Seattle, where he joined the faculty in the bioengineering department at the University of Washington. In 2008, he assumed the title of chair of the Department of Bioengineering and continued to serve in this role until 2013. He is currently a professor in the Department of Engineering, as well as an adjunct professor in the Departments of Chemistry, Oral Biology, and Global Health. Dr. Yager has mentored dozens of undergraduate, graduate, and postdoctoral students. He has also received numerous honors and awards, including a Top Doctor's Award in Global Health in 2012. Dr. Yager has extensive grant funding for his multitude of research projects, and he has published over 200 articles. He also has patents on over 30 devices. He speaks locally, nationally, and internationally, and has done so for over 3 decades. Some of you may have seen in November of 2014 his talk on TEDx Rainier. So please join me in welcoming Dr. Paul Yager as he speaks today on disposable microfluidic devices for point-of-care diagnostics, pushing out the boundaries of when and where we can detect pathogens.
Thank you. I think I can get rid of this one. Yeah, thank you. Thank you. I really appreciate the invitation. Although it was a little dark when I got up this morning. I'm just not used to doctor's hours, I confess. I'm the wrong kind of doctor. Let me quickly pull up. Let's see. Yeah, okay. So I hope this works. I first want to say thank you to a lot of people. I've been working now for about 20 years on the idea that we ought to try to decentralize testing, particularly for pathogens, and that it could be done, that it would benefit people in the U.S., and then 15 years ago, the lightning struck. Someone took me over to PATH, and then I learned about the fact that there was a world outside the United States, and I've been thinking a lot about global health and how we could particularly aid people where they don't have the resources we have here. So most of what I do is talking about developing technologies that could work here, but could also work perhaps even with greater advantage in the developing world. I want to thank these 2 people, Barry Lutz and Elaine Fu. I've worked with them for about a dozen years. Barry is currently a assistant professor at UW, and Elaine Fu is a faculty at Oregon State right now. But we work very, very closely together, and most of what we're developing was co-developed with those 2 people. Right now, I've got a fairly large group. That's actually, the picture is small, the names are right, and I'm thanking these people. Many of them did a lot of the work that I'm going to show today. So the problem we're addressing is the fact that you have these big, lovely machines available down the hall here that do a fabulous job of taking biological samples and extracting information, particularly presence of pathogens, using immunoassays and nucleic acid assays. We're very, very good at that. And the world is converting to those technologies. It's just that not everybody has access to that kind of equipment. And even if they had the money, they have no place to put it. And that's true whether you're talking about labs in the developing world. It's certainly true if you're talking about taking the technology home. There's just no room for the footprint of the technology to do that, and no need. You don't need something that can do 100 tests a day if you only need 1. So we're trying to find out what's in between. And the model for this really is what happened to computing since the 1970s. When I was starting graduate school, if you wanted to use the computer was a place at the university, and you brought punch cards to it, and then you got your printout, and you walked away. And that centralized model is dead, and now we have our cell phones that have far more compute power than that central computing facility ever had, and it's a lot more convenient to use, and then we built things around it, like the internet. So we have some amazing abilities to move data around. However, when it comes to medical data, it's still largely collected at places like this hospital we're in, which is fine, and our model has been to see whether we could use the cell phone network to decentralize the data collection and, frankly, use the phone, this object that sits on our hip, as the hardware that we would need, and that we try to minimize the amount of hardware, ideally have something simple and disposable that goes along with that. And that's the model for everything we're doing in the lab now. And it may not be the only model. It's certainly not the one that's commercially active, but we're trying to, please forgive me, Hans Kuhn, to shift the paradigm over to a technology that might be somewhat different way. And I'll hopefully we'll have a lot of time to do Q&A on that, because it's an interesting area, this issue of what the paradigm would be in this model. So if we can't bring the person to the lab, bring the lab to the person, wherever that person is. We're very interested, however, and I say this not just for you because there are MDs in the audience, but because we've talked to the FDA, we've talked to various people, and the idea is not to take the testing away from the medical community and the healthcare community, but to actually extend the reach of the healthcare community so that, in fact, clinicians have the ability to track patients over a wider area and make sure their patients are healthy in places where we have an infrastructure and create an infrastructure where no infrastructure exists at all at this point. If you travel and you go interesting places, you begin to see homes that look kind of like this. So you realize the definition of home does not mean it's got a refrigerator and 110 volts AC in the. You can have situations that are extremely dire. So when we think of what storage conditions we want, it's 40 degrees C for a year, okay, with no power, no refrigeration, no nothing. Just that's all you have, and that you should be ready to go with a test under those conditions when you need it, where you need it. And then get the information to wherever it needs to be sent. The World Health Organization has this mantra called the assured criteria. It was actually developed in collaboration with King Holmes. One is, the acronym stands for A for affordable by those at risk of disease. And they're obviously thinking about global health, but it's really applicable elsewhere. Sensitive, that means medically sensitive and specific. So you'd like something that's as good as laboratory-based tests if you can get them. A new criterion is user-friendly. That is one that doesn't normally exist for laboratory tests because either it's in a machine or you have somebody with a highly trained lab med degree who knows how to use this thing. We're saying none of that. Keep it as simple as possible. Rapid. Now, rapid could mean if you're going to see a clinician, the clinician should get the test done before that patient leaves the face-to-face interaction with the clinician. Now, how long is that? Well, maybe from time where they show up, it's less than an hour. 30 minutes comes up as the magic number a lot. We'd love to do everything in under 30 minutes. Sometimes it's a bit tricky, and we'll talk about that. Robust, and like I said, storage conditions as well as robust in use. You ought to be able to drop this thing, whatever it is, and have it still work. A major goal of WHO is equipment-free. That's because the model that works in the developing world is the lateral flow test. And for us, that's the pregnancy test kit. That is the model that works around the world. So that's the paradigm they would like to pursue. We're trying as much as possible to get to that point, and we'll show you where we're going. And then finally, the really hard part, and I could spend an entire lecture on this, is delivered. That's the tough part. It's how to get it out the door to people. It means finding commercial partners, getting them to manufacture it, getting it delivered, sold, and emphasis on sold, because if there's no money transferring hands, there's no interest in people from making it. So this is the part that I get into. I did give you the disclaimer slide. I have no financial interest in this at this point. I hope the university has a financial interest in soon, though. We'll talk more about that later. So we're trying to keep it simple. So this is one of my graduate students who's now going to demonstrate what we think the end user should have to do, which is open a pouch, a foil pouch, to keep it dry and stable for a while. Pull out at most two things. One is the device, and one is a sampler. In this case, the sampler is the nasal swab, because we're working primarily initially at upper respiratory infections. We're looking at MRSA infection and RSV as viral and bacterial diseases. Insert the swab, rotate it a few times, and close the box. At which point, the patient does something else, or the person, whether it's a patient, whether it's a lab tech, and that's it. And everything is going on inside that box that you took out of the pouch. There's no machine, there's no power, everything's inside the box. We'll tell you about what happens inside the box to close it up. And then, all the patient might do is either look at the readout, or, if you want to stay attached to the system, take out your cell phone and take a picture. And that picture contains all the data that you need to send the data anywhere. Why pictures? Because most of us have cameras on our cell phones, and that's rapidly becoming the norm in the developing world as well. It immediately puts you in contact with the rest of the planet for sending out either the data that you could read, or it could be encoded. So the patient doesn't even have to know what the results are, because you can essentially encode optical data so they don't know what the meaning is. So that's the model. It's not meant to be necessarily as simple to read as a lateral flow test. And then once it's out there, of course, the physicians can get access to it. The medical records can be tied in. You can send people, send an ambulance directly to the person if you like, or tie this into clinical research and actually run trials of new tests on the device in parallel. The core technology we've gone with since about 2008 is different from what we've done for years. For years, we worked on what I'll call classic microfluidics, and we sort of created a whole bunch of buzz and industry, and probably some of you already work with devices or have tests done on devices that incorporate microfluidics. What we said was, let's keep it as simple as possible. Let's cut away all the instrumentation. And the simplest way to do that was to use wicking to move fluids. And that's what makes lateral flow tests, things like pregnancy test kits. There's a sample pad, there's some chemistry, there's a wick made of nitrocellulose, and another one that sucks all the liquid through. And you can throw away the instruments and just read the results. And they're great, but they're a little simple. They don't actually do a lot of complex stuff. So our argument was, if we could do this in 2 dimensions and don't bother reading this slide, we could actually do things that are more complicated and sequence different types of reactions. So the thing to see is this cartoon. This is a device we built about 5 years ago. It has paper. It's got a nitrocellulose main path over here. There are 3 pads that have food dye on them, that's all. And then another wick that's meant to suck up the extra water. And it's sitting in a little trough about 2 millimeters deep, filled with water. This isn't what we actually build for the developing world, but it was the demonstration that got us, frankly, the money to go on with this, because the idea is simple. So what you do is you dunk in your device into water. That's how you activate it. Oops, sorry, it should start. And when you stick it in, water begins to dissolve reagents. The reagents then move sequentially to a spot over here. And this is what you do if you were doing a variety of a lysis test, for example, or a variety of reactions. You bring reagents sequentially across a particular zone where you've got capture on it. And that sequential rinsing and passing of reagents across allows you to do sequential chemistries. Now, nobody's doing anything except sticking it in water. The water's draining. We have things at different lengths, and the water is simply being turned on and off by the paper structure. So we call it programming in paper. It's very simple. It's ridiculously inexpensive. And that's the approach we've been taking as much as possible to get these sorts of things low enough in cost and robust enough they could be used in the developed and developing worlds. We don't just use paper. We often use the things that are in lateral flow tests. They range from cellulose materials to glass fiber to nitrocellulose. And these things are fairly well engineered in that they've been developed for commercial products for a while. So we have that amount of regularity. You can see these things are, of course, very highly irregular actual physical objects. So we have to engineer around the imperfections in the systems. Just as a small example, you can develop switches. Just as an example of things you can do, this is an all-fluid system. The idea was have something that moves a liquid from the bottom to the top in stages. We have valves, and the valves are actually sponges. Sponges expand, they push things across, it opens up channels, and only when the liquid opens up the next sponge does it move there. You can see the red and liquids don't mix. It's made with a couple of thin layers of plastic. So this is the kind of valving we want to use, where we program in the timing of these things without having to actually have someone control a valve whenever possible. So we got 2 big programs. I'm going to really zip through one program in a minute or two, which is this one. It's an NIH-funded project. The aim was to make a more sensitive influenza test. Current influenza tests are not adequately sensitive in the U.S. population. They actually work fine in Japan. That's a whole other story, separate lecture. But they don't work in the U.S. So NIH funded us for a nicely funded program to do work on developing a more sensitive immunoassay. And it has, among our various partners, PATH, Seattle Children's, GE Global Research Center, which makes the papers, and David Baker's group in the Institute for Protein Design. And that's been an enormous amount of fun. And I'm really only going to focus on that for the moment. We're working with his binder molecules, which are designed to bind to different parts of the influenza virus. Primarily, he's been attacking the HA antigen, the one that is responsible for attachment to cells in the respiratory system, and is therefore a good target for an intervention. What we're doing is taking the crumbs off the table. The things that don't work well as therapeutic proteins or therapeutic binding molecules actually can work fine as diagnostics. We just have to kind of hold them onto the paper on one end and then get a label on the other end. So we are making sandwich immunoassays with this sort of technique. We have parts that are binding to the hinge region, the influenza, which is highly conserved, and then the highly variable head group region that includes the sialic acid binding sites. Whole other lecture in detail on this. To make things amplified, we're using chemistries that allow us to make an antibody or protein binding event more visible. So typically people in historically used gold nanoparticles in lateral flow tests for like the pregnancy test. That's the purple color you see. And you can make those particles bigger and much darker by adding chemistry that adds gold to them. Or you can use proteins on the system that actually would convert colorless substrates to very dark substrates. And for the demo, I'll show you we're using the color one. It's actually gold added to gold, which makes larger gold particles. This was done by Bhushan Toli, who's become our wizard of valve fabrication. And this is inexpensive, if complex looking. And what he's done is boiled it down to a capture spot and a control spot, all you really need for the test. It's a gold assay using the gold stuff over here, and it gets there first, so you have gold labeling. There actually was a sample that went into water, only two. There's the gold itself. It's a kind of faint spot. If you keep going, the amplification first he rinses, and then the amplification reagents get there, and notice that spot over there, the analyte capture spot, gets a lot darker. It's dark enough by a factor of 10 that it actually enhances the LOD by about a factor of 10. That was what we were targeting. We'd like to get to 100, and that's kind of where we're working for the next couple of years of the grant. I said there were two projects. That's an extension of one. It's sort of a quantitative improvement of what we can do with immunoassays. No doubt at some increase in the cost because of complexity. The big leap that DARPA is funding actually is a nucleic acid test at remote point of care. Because today those things are all big instruments that you plug into the wall. They take a while. They're expensive. And are largely inaccessible to people in the developing world. And we thought that was the really big leap. To be able to get developing world people out in the field at level 0, if possible, in homes in the developing world, have access to the same high-quality, extremely sensitive, and extremely specific tests you could do in laboratories. So there's a cartoon. We call it MADNAT for multiplexable, autonomous, disposable nucleic acid amplification test, and I'll say MADNAT a lot, so I apologize for that. Initial targets were MRSA, and actually, while it was originally influenza, it became RSV. Working closely with people in Ferric Fung here in lab medicine, as well as Jan England at Children's to work on the respiratory work. Large program, a lot of people funded in multiple organizations. Again, same cast of characters except adding Ferric Fung on this one. And Epic Bioscience is a local company that we worked with in the past that does nucleic acid probes and primers. So the cartoon for this that got us the money was this idea. Suppose you have a swab, and the swab's got green stuff on it because it's been in your nose. And you have a device, you insert the swab into a tube, you swirl the tube and another one full of buffer gets injected into a fluidic circuit, and then the circuit wets, and then at some period later you have a series of dots. You take a picture of the dots with your cell phone. And by the way, I love doing 3D models because you can make people believe there's something that could actually happen no matter how much simplified it is from what you actually get. It's also fun for me to take a break to do it. But so we've been funded by DARPA to do this. Everybody knew this is a big stretch because no one had done anything quite like this up to this point. Again, paper-based, so the guts of it is all paper, and I'll show you how that is. There's stuff wrapped around it that isn't all paper. Multiple pathogens at the same time, so the aim was to multiplex by either DNA or RNA. So we have to include both an amplification step as well as a reverse transcription step at the beginning for the RNA targets. Any place, simple for anybody to use it, and low enough cost, and this is the interesting question. Everybody says, how much will it cost? That's a dynamic question, and we can talk more about that. Hopefully, less than it costs right now to get an HIV-tested Bartell's drugs down the hall. That's the hope. Again, cartoons, they're great cartoons. I'll show you what it actually looks like now. So what goes on inside the box is really 4 things. One is a swab transfer that starts outside there and moves the sample into the box. We've stuck with swabs because it was simple and it's straightforward, and we'll see that we're going to keep going with swabs for a while. And those could be from the nose or any place else. We then get maybe 1 to 10 to the 4th copies of a sample into the device on the swab, at which point we have to lyse the sample. We have to break open viruses or bacteria and maybe reduce other debris that might be in there. Then we have to do an amplification step. And these amplification steps, if you haven't actually done this, basically means allowing enzymes to work for a while. A combination of enzymes, the NTPs, probes, primers, that chug along and make many, 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 many copies of the original target DNA or RNA, but only the target and not any of the rest if things work perfectly. When you're done, you should have on the order of 10 to the 12th copies of what you started with. That amplification process takes some time, and our job is to keep the time as short as possible. People usually think of it as about an hour. We're going to much shorter times. And then have something that's, again, eyeball visible, or it could be picked up by a camera. And that's all we're doing with the box. Swab transfer is really simple. Sorry, I didn't need to do all that part. I just discovered a bug in my animation. The swab goes into a tube, and inside the tube, we don't have to measure, no complex user steps. It's just what you saw with the end user earlier. The lysis step gets more interesting, and there are a couple of ways to do lysis. Some of them are completely inappropriate, or you wouldn't want to have the chemistry that people use in the laboratory accessible to your 3-year-old who might grab the test, break it open, and eat it. So we had to think about chemistry that was relatively safe, and we focused on an approach for an enzymatic mixture. It's called a chromopeptidase. It actually is very effective at lysing both gram-positives and gram-negatives rather rapidly. In fact, in many cases, in a few seconds, you can break open Staph aureus. So we were really impressed with this. But then you're stuck with all the cells, all the proteins, as well as the enzymes itself, some of which are proteolytic, and they would kill our downstream stuff. So we had to then do an inactivation step, which we do very simply by raising the temperature to 90 degrees for about 2 minutes, and that's all you need to do. So there's a heating step that has to occur in the box as well. That leaves you with nucleic acids ready to go, which is fun. So far, a chromopeptidase works on a broad range of bacteria. The only ones it didn't work with were mycobacteria, and because of TB, we were really interested in doing what we could with mycobacteria, because for global health, that seemed like a really important target. So this is not going to work for mycobacteria. I'll get back to that in a second. To heat it up, we literally use a very simple technique. It comes from the chemistry from the meals ready-to-eat stuff that the military uses. It's taking little beads of magnesium and iron. You dunk those in saline solution. They rapidly corrode, and they corrode fast enough and generate enough heat that you will boil the water around them. So you can actually generate steam. You get to 100 degrees very quickly with that water. That's our heat source. It's really cheap, thanks to the MRE stuff. So we use that to heat, but that will get you rapidly to 90 degrees, and that's how we do this one. And it works really well and very rapidly and cheaply. It turns out to get the lysis for tougher things, and we're trying spores now. We did a simulant for Mycobacterium tuberculosis. It's Mycobacterium marinum we can use in BSL-2. And it's really simple. It's a glass bead with, or rather it's a vial, plastic vial with a few glass beads in it, and then a rotating spherical magnet. The magnet costs a couple of cents. We can put a wire coil around it, so basically we have an alternating magnet in which the field flips the ball up, down, which turns into rapid rotation. It appears to grind the bacteria. It's like a bead blaster, but with very, very low energy, and it can run literally off the audio output of your phone or any sort of audio player. That's all it takes. You just put out a 30 hertz-ish signal, so all you hear is a hum, and in 10 minutes you will chew up your bacteria to nothing. What we love about that is it's dirt cheap, takes really little power, and you know that your audio player typically says it will run for 3 days playing. Well, that's how long you've got out of the power in your audio player or your cell phone. So it's really something that's applicable any place. And it gives you a front end which will chew up the mycobacteria very effectively with no chemistry whatsoever. So it's really very nice. So this is something we're actually thinking of putting into the devices if they're meant to be used with mycobacteria. Alternatively, you can make a bank of these things and do a bunch of tests in a laboratory for cheap without complex chemistry. So we like this as a generic technology. It works. It's not as efficient as bead blasting, but it takes a little longer, but it works, and it works quite effective, so we're really happy with it. So we got a way to access that. We're going to be looking at anthrax spores, or actually B. cereus the next little bit, just to make sure that B. cereus spores could be equally well lysed. We think they will, but we don't know how long it'll take. So amplification. This was actually the hard part. This is the part that people didn't think we could do, and the question was, could you amplify in a paper matrix? And that was our first 18-month challenge. We got it in about 6 months. We wanted a true isothermal method, so we didn't have to do with PCR this thermal cycling. That's time-consuming and costly to drive the temperature up and down, up and down. So fortunately, there are about 20 methods now available that allow you to amplify at a single temperature. It's a balancing act of enzymes and probes and primers, and it takes a little bit of computation to do it. But when you've done it, you can actually get a system that will amplify 10 to the 12 copies from a single copy relatively quickly. has to be fast, sensitive. We thought 100 copies. We're actually, as you'll see, able to do much better than that. DNA or RNA, so we have to have, ideally, a one-pot reaction that would allow us to put in an RNA sample and get out 10 to the 12 copies, or DNA. And we wanted the output to be detectable by a lateral flow strip. Very simple primer design. Turns out there's a method called isothermal strand displacement amplification that had been developed by a company in San Diego, which was purchased by the company Elitech that they're working with. And their method's the one we've been using for the last 3 years. It works. Down here is the time course for amplification, and you can see that at 10 minutes you actually begin to see products, and it's actually done within 15 to 20 minutes. So we can contract our time frame for the amplification step to about 20 minutes total. We're giving it a little bit longer in the devices to make sure it works, but that's our goal, is to keep it to 15 to 20 minutes tops for the amplification step. It isn't a very clean reaction. You can see here that if these two are the actual product you wanted, there's a whole bunch of other stuff made by the amplification reaction, so we have to be selective in what we detect. We can't just detect DNA total, because there's a lot of junk that's there, even if we don't put in a sample. So we have to have a sequence-specific detection system, which is part of how we do this. But it works, and it's not that expensive. I'll skip this. It turns out there are lots and lots of products, and we have to begin to understand just something about this complex mess of all the different things it produced. So we have a whole group of people who are doing nothing but thinking about amplification technology and how to improve it. It's a real art form, so we're not just people who make things in paper. We also think an awful lot about the amplification chemistry. And I'm going to skip the mechanism, except that to say that if it just made DNA, it'd be great. The problem is that then you'd have double-stranded stuff, and there's no way for us to easily detect a specific double-stranded product. So we have to shift the reaction so that one of the two products is made, one of the two strands is made in excess. So you use asymmetric primers, which gives you something you can grab hold of. But the result is you heat, run the reaction at one temperature for a while, and then you detect at room temperature. And when you do that, not only can you see this amplification that's fast, there's 15 minutes done, and this is 120, 60, 30, and 10 copies over there. This is using real-time detection, a conventional instrument. This is lateral flow detection, where we put one capture primer on the paper, a detection primer, in this case, on a blue bead, and your amplicon in the middle, and you can see you're actually detecting down to single copy, which is where you begin to get the dropouts. So that is great, because you either have 0 or 1 copy of this thing. So this is as sensitive as any laboratory test you can get. Now, these are run in a tube and then detected on paper. So our job has been to get the detection in paper as sensitive and as loss-free as it is in tube. And that was the first few months. How do we get that to work? Well, now we need it at temperature, not at room temperature, but we need it actually 50 degrees C for the amplification. So it turns out you know how to get 100 C. And then we combined wetting the magnesium iron stuff with a wax. And the wax melts at 50 degrees. So you melt most of the wax, use just enough heat in to melt most of it. The wax then goes to 50, and then it stays at 50 until the last bit of wax has refrozen. So that's a way in which you can get a very constant temperature. So here you can see temperatures of actual measurements, and this is starting at 15, 30, and 40 degrees C, which was really, really important when we said we want to do this in the home or developing world. We couldn't assume that your starting temperature was 21 degrees C like it might be in here. And it turns out this wax method is a very good way to do that. So we spent a lot of time doing thermal engineering for how to get the temperatures right so that your wax will get you just that temperature for long enough to get the amplification done. Cost of heating reagents in this case is 10 cents. So this is also an inexpensive method. And believe me, there's actually been engineering of the wax. And the part that's amazing when you think about it is the wax now has graphene in it. Okay, and you go, graphene? That's like really cool high-tech stuff. Well, the graphene is a really good thermal conductor, and it makes the temperature more uniform throughout the wax, which turned out to be one of the strange little problems, how to get the heating distributed throughout the wax rather than occurring in one corner. So this has been done jointly between my group and PATH, and this has been really elegant. They've been working on this idea of isothermal amplification reactions for a developing world for a while. This is bringing that into a format that would work with our particular system. And I don't want to spend time, they're now square little boxes with insulators around it, but they work, and they work well, and they're inexpensive. Temperature profiles then, this is the heat, this is the lysis heater, and this is the temperature of the amplification heater. It can hold out to 60 minutes the way we're doing it now. We only need the first 25 minutes or so we can actually simplify things down to reduce costs, reduce materials. It works pretty well. The variance isn't too bad. Obviously, you need to have it fairly tight for these things to work, but we're very encouraged that it's going to work well. So finally, there's detection, and we're using lateral flow in this device. We've actually switched from blue microparticles down to gold nanoparticles, the same gold stuff you use in conventional lateral flow strips like pregnancy test strips. It works really well, and it's relatively inexpensive at the levels we use it at. So how many lines would you have per strip? Well, probably 2 or 3, because it turns out the single pod amplification reactions don't multiplex really, really well. We're never going to be able to put 15 different targets in a single reaction. So what we do instead is we split the pot, we split the sample into multiple reactions and detect them in parallel. So it's paper strips. We just make more paper strips and line them up behind it. We're modeling it now with 2 being an example of the smallest number of many we can get is 2. So we're demonstrating with 2. The box looks more like this now, and I'm going to skip through what we're promising to do. We have what's called a TRL5 demo we had to do. We had to do it actually back in July. We aimed it at the MRSA test, which was the first one we were working with, and eventually it'll be MRSA plus this. Sample was a swab. Swab had about 10 to the 3rd bacteria on it when we started out this thing at one point. That means there are more copies of the target than that, but it was 10 to the 3rd copies. This is a video of a movie that we made in July 25th, and this is the one we showed to our friends at DARPA. So here's Lisa Lafleur, super postdoc, taking a cap off, inserting the swab, rotates this for 10 seconds. So the first time, by the way, was a cartoon with the person. Basically, there was no real thing. This is the actual assay as it occurs, and there's a timer. You can see you're at 16 seconds there. Which point you close the box? Closing the box actually starts a variety of things. First, the cell lysis and the 5-minute heat inactivation. There's a valve that opens, which moves things into an amplification zone where it picks up dried reagents, which includes all the reagents we need to do the amplification reaction. That chugs along, in this case, for 30 minutes, just to be sure. Okay? Second valve flows things towards detection. We're at 37 minutes now. And then if you look real carefully, right about now, you'll see lines, you'll see liquids moving up through those 2 strips side by side there. And if you look carefully with the camera, you'll actually see, placing, in this case, an Android phone on top, taking a photograph. That photograph actually has those lines on it, which were a positive at 10 to the 3rd. Not as dark as we ultimately like them, but that was back in July. We'll tell you more about it. And then we actually did analysis, in this case, off the phone. It could actually occur on the phone. But one model is you take the picture and you send the picture remotely, and the analysis goes elsewhere. The alternative is, these days, these phones can do all this stuff as fast as a PC, in some cases faster. But here's the actual, here's the actual run of the code itself, which takes a couple of minutes, and this is in analysis. This is stuff that our undergraduates are doing. So this is undergraduate level data processing, and it's looking at images, looks at the images, analyzes them, and then eventually says, yep, those are lines, and that's where we are. So okay. The result is positive. Twice, actually, in this case. And I don't want to go through this sort of stuff, but just to say, I want to show you what goes on in the guts real quickly, because it's kind of cool, and it's where most of the engineering has been, which is to make the paper work really well. We discovered early on, it's something I was sure would never work because of my experience in flow and ducts, works in paper really well. You take a couple of tubes of liquid and you simply place them on top of a piece of paper, in this case nitrocellulose. They will drain sequentially. They will not drain all at once and make a bunch of lines. The yellow liquid drains first, then the red liquid drains, and then, and only then, does the green liquid drain. So you can actually send different fluids out there by loading them up in liquid tubes. And we said, whoa, that's different. We can file a patent on that, and that's one of the pieces of technologies we have that we're using in the system, because it very conveniently means if I load up those reagents, I can then sequentially deliver them very effectively in a very robust manner. There's a movie of it actually happening here. So here's the 3 liquids, yellow, red, and blue. The yellow one, it just runs out. You see, oh, the yellow liquid's gone, at which point the red one starts moving up. And then the red one, you'll see liquid there looking through the hole in the top of the tube. And when the red finally gets down to paper, so it's gone, there goes the blue one. It takes off. This happens. It's very reproducible as long as the paper's good and your mechanical seals are good. We spent a ridiculous amount of time worrying about how best to attach a tube to a piece of paper. So here's the inner workings of the thing, just so you get an understanding. We're only using 2 tubes, and I'll show you how several things have changed, but the general concept was true. So we have paper. Paper, actually, in this case, is fiberglass and nitrocellulose. We have swabs. A couple of valves. They're originally mechanical. They're no longer that way, but I'll show you what they look like now. We put our amplification dried reagents someplace. They're actually no longer in exactly that spot. Learning how to dry down enzymes and nucleic acid probes and primers and have them survive at 40 degrees C for a year is not a trivial challenge. Fortunately, we have about 15 years of experience in that. So it's actually pretty good. In fact, some of these methods are so good, I don't see why they're not being used for stabilizing vaccines at room temperature, and that may be somewhere we'll go with this in the long run. The detection reagents are elsewhere. They sit afterwards, and everything is pulled along by wicks. Why two? Because we're going to do, ultimately, RSV and MRSA with the DNA and RNA tests. Two solutions and a heater. There's actually a heater around the lysis buffer. So in goes the swab. After it cooks for a while, it heats to deactivate things, at which point we open the valve and liquids start flowing down from those two things into the paper. So remember, this push buffer on the back end this one doesn't go anywhere until this one's drained. So it's draining, it's picking up reagents here, going under the heater, at which point we turn on the heater. And the heater runs it to about 49 degrees for about 25 minutes. We then open the second valve, and we'll talk about how that happens, which then runs the amplicon solution into the detection zone, picks up the right reagents, and you get your lines. And that is literally all that happens. 46 minutes was about the time for this part of the reaction to work. And this is great. And I told you all about those chemical heaters, and then I lied. What happened was, a month before the demo in July, my engineers, many of whom were electrical engineers, came to me and said, we love the chemical heating, but it's taking us too long to work out the timing for the controls. It could be done, but it will take us another 3 months. We wanted a demo in July. And they said, will you please let us convert everything to electronic controls? I looked at them, and they looked back at me, and I said, are you kidding me, right? And they said, we can do it. We can do it in 2 weeks. I said, they're engineers. Okay, so I gave them what they wanted, and they ran out and redid everything electrically. So there's now an electrical heater for the lysis. There's an electrical heater for the amplification, and there was electrical actuation for the valving, and I went, you're kidding me, right? But they ran it off 2 AA batteries, and there was enough power in the AA batteries to do the whole thing twice. So they had redundancy for this, and they were able to build the devices within 2 weeks. This is the beauty of 3D printing and these sorts of things, and people who understand how to design circuits. We actually got it done, and that demo I showed actually had all electrical parts on the inside. Now, that does not mean we're giving up on the chemical heating. In fact, we are absolutely producing them. But we now have 2 lines, an electrical version and an all-chemical version. The electrical version has the downside of having 2 batteries in it, so it might go in our trash. But if you wanted to burn things directly as they were, you'd want the all-chemical version. And you can think of a lot of things where you have biohazardous waste, depending on what the samples were. We'd want to burn it as fast as possible to ash. So that's actually the design we're thinking of for really scary diseases and things outside the home, where someone other than the patient, him or herself, is handling the medical waste problem. Okay? So we're pursuing both. The current guts have been simplified a lot. We actually don't have pads on this end. We're actually soaking the strips in the chemistry, so we have side-by-side the DNA detection and the RNA detection to do 2 separate things. We split the sample. This part is pretty much as I showed it to you. The valves have actually changed. We're now using, instead of mechanical valves for this thing, we're using wax. Because it turns out you can put wax in paper, it will block liquid flow, and then if you melt that wax, and we know 2 ways to do that, electrically or with the chemical heating, you can melt it to about 60 degrees. It doesn't kill the chemistry, but it moves the wax out of the way. It spontaneously balls up elsewhere, allowing the liquids to go through. So we have one more heating step there to actuate the valve, at which point everything goes. So we've stripped it down to very simple things. There are now 4 places that are heated. It turns out we have a thermal valve that controls this as well. So it's all done by heating 4 things, which can be done either with chemistry, which we're working with PATH to develop and to work out, or electrically. And we could put in the audio lyse thing, the rotating magnetic ball, if we wanted to do things that are harder to lyse that don't work with ACP. The housing shifted a little bit. Strange little details. Here's the paper. The paper's at an angle. Why is the paper at an angle? Because if I take my cell phone out and hold it flat on this thing, and you have a flat surface, you get a really bright reflection. Like, try taking a picture of yourself in a mirror sometime or in a window, right? You get this huge flash. This allows you to use the flash and not get the reflection on the image, so you can use a standard camera and stick it right on top, and it will take a nice picture of this. Same idea, swab goes in, close the box, bend the swab, and off you go. It's just the things have gotten a little more interesting on the inside, and we've decided to store the fluids, the ones that need to go into those wells, in a couple of cut-down syringes. So in fact, we're going to use syringes, so as you move the case, you actuate the syringes, push the liquids in, rehydrate the reagents, and everything gets going. It still fits into something that's the size that you saw. It's about as big as, 4 cell phones stacked one on top of the other. And we're in the process of making these things now. These are the technical drawings. We've got shells and most of these things fully manufactured already. So what are we going to do with this? Where's the sort of the end point of all this? And there's a lot that goes into that discussion. So the core is this piece of paper on the inside. Swab access, gives us respiratory swabs. It could be other types of swabs. It could be vaginal swabs. It could be buccal swabs. Those are all potential samples. And to make the device work for a different pathogen or pathogen set, it's just a matter of changing the probes and primers. It's chemistry. The hardware should be standardized. If the swab sample is not the normal one that people would take, let's say you want to do urine, we are actually thinking about how to get the urine pathogens onto a swab. So we're actually building a small urine sampler, which could be basically a cup you would give to the patient, pee in this cup, and then when the patient has peed in the cup, out goes the swab and goes into the device. It's a simple way to use the same detection hardware, the same sample analysis hardware. And finally, for blood, we thought about sticking the finger directly into the box and pulling the finger out. Maybe. Nobody likes that. People want to make sure they get their blood, and you want to be able to express blood even if it's a finger stick. And we're thinking finger stick samples. So we're going to build essentially a small device which will transfer pathogens to the swab. Pathogens or the nucleic acids from the pathogens. That's another sub-project for the next 18 months. But keep everything as simple as possible so we don't have to change the box. But that engineering will be done, and everything is about how you get the sample into it. There's 2 versions we want to make then. One is what we call the electronic MADNAP, the electrical version with all battery control, which might be the fastest one to get to market. It might be the one that would work in the U.S. domestic market rather well. On the other hand, the chemical one seems to be the one for the developing world for a lot of good reasons. So we're pursuing that very actively in our collaboration with PATH to get all of the heating steps done using the MRE chemical heating system, and it looks like it ought to work fine. So what are the next steps? Well, we're optimally implementing this real-time one for viral samples. Right now we're working RS. We can actually get down to about 10 copies of detection at this point for RNA samples from RSV. I don't think there's any reason that any other RNA virus, whether it's influenza or HIV, couldn't work equally well in the system. It's just a matter of getting the sample in. We're working on the systems integration of these 2 versions. We're interested in, as we showed you, the optimization for different sample types. So we're going beyond the simple nasal swab sample. And finally, very much interested, although it's in gray because it's not going to be funded by DARPA, exploring the global health applications with malaria, TB, and HIV. And very importantly, that the D part in Assured, it's the delivery strategies, how to develop a commercial strategy and commercialize this. This is something that I'm spending a lot more of my time than I thought I might on these days. This is the part where the questions and answers get interesting, and I'll just try to seed the conversation with a couple of things. Who might take advantage of a fast, inexpensive, simple-to-use NAT test that doesn't require a degree and not a whole big booklet of how to operate it? Well, could you use this in a hospital ER? I think so. You needed to get an answer right there very quickly and say, whoa, does this person actually have disease X? If they'd had this in Texas a few months ago, we might not have had our first Ebola death in the U.S. You can think of that as an example of an infectious disease you might want to have a test for sitting in the ER. One small example. Small clinical labs would certainly be fine, but clinics with outpatients, so a variety of outpatient circumstances. If that patient, you could do a test on them right there before they actually left. You could make sure they got a prescription or hospitalization step right there. Physician's office labs, and even the physician, him or herself in her office, could do this test while the patient's in front of him or her. If it's done in 20 to 30 minutes, we think that might be acceptable. Obviously, a growing interest these days in pharmacy rapid care clinics. It's sort of, I know they're the enemy to major hospitals, but they're a really interesting clinical market and rapidly growing one. We want to tap into that because they seem like the kinds of places where they don't want rooms and rooms full of big machines, but they do want to get certain tests done. That could be dentist's office, for that matter. Nursing homes, and eventually home users. These are not equally easy markets to tap into, and we're very much interested in exploring which ones could be the first place we could make a product for it. It really is interdependent. We sort of, when I gave my talk at at the TEDx talk in November, I sort of tried to figure out, okay, how am I going to make this talk? And it was hammered on me that I was not talking to doctors in the audience. How do you get them interested in home testing? And I sort of ran, these sorts of questions. How long does it take me to get an appointment? There's an issue that could trigger a home test, right? This is a real factor for real patients. People who travel, there's a lot of travel medicine questions people have. Sometimes workers don't want people to come into the office if they got the flu, and that's true for workers at Boeing. It's also true for people at colleges. How many times do people have a situation where they don't, whether they've got a treatable, serious disease or it's just another cold? And if you knew what those diseases were and you had a panel, you could do a panel test of 5 to 10 tests on this kind of device and identify which one it was. Could I just go to the pharmacy to pick something up for it? Well, you need to know what it is and who's going to recommend that unless they know what has some sort of trusted positive test, and this might be a mechanism for that. Do you go to the ER when it's snowing? Think of the East Coast, and would you do that? Probably not. What's that itch down there is a really popular one where people might be hesitant to go see their personal physician or family physician, particularly if they're kids, to deal with problems, and these are real problems. The STI issue is quite real. Was an antibiotic course effective? Is it working? Okay, is it doing what you hoped it would do? You could send these home with people who are on courses of antibiotics to make sure they're actually functional. Strep test. Home strep test is actually one of the most popular ones. The biggest concern we have is which parents are willing to scrape something in the back of their kid's throat and make them gag. But it's the test that everybody likes as the clinical one that would be most useful in many ways. Finally, the one that I'm most interested in, I see Judy in the audience there, is whether or not this could be a trigger for PEP. If a vaginal swab is performed within 72 hours and you've got the idea that someone has been exposed to HIV who is not otherwise him or herself HIV positive, this would be proof positive that you needed to get on PEP rather than just saying, trust me, I think it could have been a bad event. We think that could be useful both here, and my daughter assures me this would be a useful product. I think in the developing world where the problem of HIV transmission to young women and girls is really severe, this is combined with a PEP program could be really, really effective. So this is my hope, is that something like this will actually get out there and have some impact in the developing world. At home, it's the pharmacy, and we're looking at how well the OraSure test is done. Right now you go to Bartell's and there's the group health care clinic right there in the middle of the pharmacy, and you look at CVS and their approach. This is really the way a lot of primary care is going to happen in the future, whether we like it or not. And I think it's an approach that we want to think about. So what I'm going to do right now is stop. Thank you for your attention and open it up to questions. Thank you so much. Thank you, by the way, for staying awake.
Hi. So I have a very mundane question. In developing countries, I've seen a lot of cell phones. They use them. But if people don't have electricity, how do they charge them?
These solutions have been made, and they're solar chargers. There are places that you can pay to get your phone charged in a lot of these places. But it's one of those things that came along with the phones as part of the infrastructure. Many years ago, I was in a township in South Africa, and my amusing experience was this. We were in a township, which is basically a town of 35,000 people living in wood huts. They had good medical care, and we were walking through these mud huts, basically, or wood huts with corrugated steel roofs. And I noticed there were power poles every place and wires going to these little huts. And I said, people must have power here. So I whipped out my Motorola flip phone, which is what I had at that point, and opened it up. And I looked down and I had 5 bars. I went, 5 bars? So I turned to my host and I said, do people have cell phones here? He gives me this look of mild disgust. He says, everyone has cell phones here. And most of them are much nicer than yours. So I was like 2 years behind. So the fact is that in many places, these are becoming absolutely central. Keeping them on, keeping them charged is something that is a solved problem in the developing world. Can they use them for information? Obviously, could you use them for this way? If they have a camera, you could. It doesn't have to be a great camera. They're all pretty much the same on these things, just getting more and more bits. You wouldn't have to do this for a particular market where people didn't have cell phone access. You could certainly have these things show up as lines that we're doing it now. It's just that when we talked to the FDA was extremely negative on the idea of selling another test to U.S. home users, which would allow them to go do stupid things, like not go to the doctor because they had a false negative, or go to the medicine cabinet and take whichever pills they had, whether they were the right pills or not, and take a bunch of them and kill themselves, either way. They want accountability into a test, and they really like the idea that the test in a U.S. home might occur, but the information goes to a healthcare provider who then makes the clinical decisions, and including the clinical decision of what to tell the patient back home. So the idea of moving the data out in an encoded form and then back through that loop is really what the FDA would like, and that's why we like the imaging technique as one of several ways we could encode the data.
Paul, thanks very much for an interesting talk with lots of potential ramifications. The question in developing countries, presumably in large part, is going to be cost. So how much would the device cost, and who would pay? Are you thinking about the country's health authority or the individual? And in the developed countries,
Right.
of course, the main road bump is going to be regulatory agencies and CLIA,
Right.
and et cetera, et cetera. So can you comment on both of those?
I can comment on it as well as I can. So let's start with cost. The answer is, right here, the Auric quick test sold at $39.99 at Bartell's. You can maybe get a 20% discount if you buy it on Amazon, which you can also do, by the way. So how much does it need to cost? How much must it cost? Well, someone has to make some money on it, so the manufacturing costs have to be low enough. Almost all the parts of this thing can be injection molded parts, which means the parts themselves are pennies. Right now, it looks like the most expensive costs are the reagents themselves, and we know that all those reagent costs drop with economies of scale. So if you order a lot of 1 million, you get a much better price than you do if you order a lot of 10. And right now, we're still in the research phases. And it's very hard to get a direct quote from a vendor who says, yes, if you order 1 million times that amount, I will drop the price to this, because they often don't have the production capacity yet. So there's going to be reductions in cost. The first rounds will be expensive. So there's no question that if you had an unsubsidized cost for the first thing, it's going to be bad. If you look at, for example, the introduction of the Gene Expert system, which is the instrumented DNA testing system with a box and a list price in the U.S. of about $75,000, they rolled that out in South Africa for TB testing, and there was no way they could charge $75,000 per instrument for it. So they cut the cost to $10,000 with subsidies from Gates and others. So the answer is, first introductions are frequently have to be subsidized to even determine what the economies of scale would be on the final system. So I would imagine there'd be a sort of cost kind of like that in a U.S. product initially, but for a developing world cost, you'd probably have it at about 10% of that initially. How that would settle out, we don't know. The other thing to realize, and I keep getting hit in the head with this with my interactions with developing world, and God bless the Gates Foundation for making me aware of these things. There isn't just one developing world. There's zillions of them. Even in a single country in the developing world, you may have 25 different segmented markets, from extremely rich people in India to the people who live in those plastic tarps, like I showed in that picture. So within a single country even, there isn't one market. There are multiple markets. So in fact, the developing world sustainable markets, the ones where you'd actually have, let's say, someone manufacturing this in India, would almost certainly have different pricing tiering for different parts of the market, depending on who's paying. It's not a simple answer. I wish there were, and it's not a simple test. Again, if, for example, this were to be used for a rollout for reduction of malaria, okay, for the malaria elimination, it might be coupled with a malarial elimination project. So you'd have a cost for the diagnostics, and you would make sure you met that. We don't think there's any reason that the inherent cost of goods, even in a first round with high production, should be more than $10. But that's 10, and that means if you sold it at 10, you'd lose money, or you'd break even. So there has to be more than 10 if you're going to sell it in the U.S., for example, and that's a quantity. And you're going to lose money your first couple of years no matter what you do. It's a really, really tough problem. My only business experience was association with another company that struggled for a while and now, eventually was bought out by Sony. My MBA is still in the works, let's just say, and this is a very complicated and very interesting problem, and it's global and it's not simple. Judy.
Paul, that was a great talk. Thank you. Two questions. One is, talk to us a little bit about what you're thinking about applications for antimicrobial resistance testing through this approach. And the second is, you talked about urine. The other obvious specimen that's important, particularly in low-income countries, is stool.
Yeah. Working backwards. Okay, for the antimicrobial resistance, in most cases, antimicrobial resistance is based on development of a new acquisition of a gene that has a particular trait. Those genes can be sequenced, and they are in many cases. We know what those sequences are. As long as you have the rights to manufacture a probe to that particular sequence, and often they are proprietary because people have got them, you can go and make a test which would be very specifically identify the presence of that gene. So you, for example, in our MRSA MSSA test, we have one that's for a pan-Staph aureus gene, okay, the LDH1 gene, which is typical of all them. And then we have the MEK-A gene, which is a bacterial resistance gene. And we're doing exactly that. So we test for two genes simultaneously in the sample. It's a complicated problem because it turns out that other bacteria share that particular gene. So if the ratio isn't 1-to-1 in quantity, you may have Staph aureus that is not, resistant, and you have another strain that is. So it's not a trivial thing to do this. There are lots of schemes that have come up for doing that. Sometimes you have to 2 or 3 or 4. But the answer is, we think we could easily do 6 parallel lines. We've successfully now triplexed one of these things using some recently in the last couple of months. So in theory, you could do 18 different targets. More likely, it would be 12 with internal controls. So 10 to 12 is good. So you could have the base organism and 1 or 2 different resistance genes on the same strip or in different strips to know whether you have that resistance. So for microbial resistance, that's one thing. We actually were involved in a project that PATH led, and Micronics, our former startup company, was also involved with, called NIAID, and it was to look for, they're called Schedule 1 pathogens from stool. It was meant to look at biowarfare agents because that's where the money came from for NIAID. Stool samples turned out to be challenging for nucleic acid amplification. And I'll just say that. That's the politest thing I can say, but it was challenging at every level. One, it's such a variable sample depending on what people are eating that day, how sick they are. We got almost most of the way there, and there are some successful stool-based tests out there now, and I would argue if it can be done in another format, it can be done in this format. So I don't know of any reason we have a problem. Heme, for example, is classically a problem for PCR, and you have to get rid of heme. This amplification reaction is much less susceptible to heme problems. Heme is an issue, obviously, in stool, particularly for sick people as well. I think we can nail it. We have a couple of approaches we'd like to try, but we have not actually proposed it. DARPA wasn't particularly interested in stool samples, so we don't have funding for it. It's definitely a direction we could go. We just have to get lab techs with, what's it called, the anosmia, where you can't smell, to start there. But it's doable. Yeah. Thank you. Thank you.